Hello students, welcome to this session. So let's study methods of anaerobiosis and non-sporing anaerobes. And first we will see methods of anaerobiosis. So anaerobes require anaerobic environment, that is absence of oxygen environment. So there are few methods. So they are called as methods of anaerobiosis. So they are vacuum desiccator, candle jar, McIntosh Fields jar, anexomat, gas pack system, anaerobic glove box, and RCM or thioglycolate broth. So RCM means Robertson cooked meat media or thioglycolate broth. So first method is vacuum desiccator. In this, production of vacuum is achieved by incubating cultures in vacuum desiccator. It is not effective method, so it is not used, and plus there is risk of explosion, so routinely it is not used. Second method is candle jar. So in this, inoculated media are placed in a jar with a lighted candle and the jar is sealed. The burning candle reduces oxygen and flame goes off. This provides atmosphere of 3 to 5% carbon dioxide, and this is useful for capnophilic bacteria. So what do you mean by capnophilic? So bacteria which grow in presence of 3 to 5% CO2, they are called as capnophilic bacteria, and example is Brucella. In this diagram you can see that jar plus inoculated plates and then candle. Now next method is anaerobic jar. So principle of this is displacement and combustion of oxygen. So first step is evacuation of air, that is done by outlet tube connected to the vacuum and then air is removed. Then second step is replacement of hydrogen. So that is done by inlet tube connected to the hydrogen supply cylinder. And then removal of oxygen is done by use of catalyst, and then reaction is carried out. So O2 plus H2 combine and form H2O, and then in this way the oxygen is removed. Now next method is anoxomat. So this anoxomat is automated equipment for anaerobiosis. It evacuates air from jar and replaces hydrogen from cylinder. That is done automatically. So now next method is gas pack system. So principle of this is absorption of oxygen by chemical method. So packet of gas pack which contains sodium bicarbonate and sodium borohydride is kept inside jar and water is added and lid is closed. Next method is anaerobic glove box. It is anaerobic incubation system. It provides oxygen-free environment for inoculation and incubation. It is fitted with airtight rubber gloves to insert hands to work. Next method is Robertson cooked meat media. So principle of this is reducing agents like glutathione, cysteine present in meat utilize oxygen and it provides anaerobic environment. So here you can see this Robertson cooked meat broth which contains nutrient broth with pieces of fat-free minced cooked meat of ox heart. Let's study non-sporing anaerobes. Now we'll see non-sporing anaerobes. So anaerobes, they are bacteria that lack enzymes, superoxide dismutase or catalase or both, and makes them susceptible to oxygen-derived free radicals. They obtain energy from fermentation process. Non-sporing anaerobes, they are anaerobic bacteria that do not form spores. The habitat of non-sporing anaerobes, they are present in normal flora of skin, mouth, mucous surfaces of respiratory tract, gastrointestinal tract and genital tract. Now classification of non-sporing anaerobes. So they are gram-negative bacilli. So in that bacteroids, Privetola, Porphyromonas, Fusobacterium, Leptotrichia are present. Then in gram-positive bacilli, Eubacterium, Propionobacterium, Lactobacillus, Mobiluncus, Bifidobacterium, Actinomyces are seen. Then in cocci, Peptococci, Peptostreptococci, they are gram-positive cocci, and gram-negative cocci, Veillonella. And in spirochetes, Treponema and Borrelia. So these are the examples or classification of the non-sporing anaerobes. So here you can see common locations of infection involving anaerobic bacteria. So there are brain abscess, chronic otitis, mastoiditis, sinusitis, thrombophlebitis, pneumonia, abscess, peritonitis, appendicitis. You can see dental infection, cellulitis, then diverticular abscess. pelvic inflammatory diseases. So these are all, or endometritis. So these are all common infections caused by the non-sporing anaerobic bacteria. Anaerobic cocci. So Peptococcus and Peptostreptococcus usually produce mixed infections along with Clostridia or anaerobic gram-negative bacilli. So it causes puerperal sepsis and other genital infections, wound infections, gangrenous appendicitis, urinary tract infection, osteomyelitis, abscesses in brain, lungs and other internal organs. Now we'll see few gram-negative anaerobic bacilli. So one is bacteroids. So it is most common anaerobes isolated from clinical specimens. It is pleomorphic capsulated and it grows on brain heart infusion agar in 10% CO2. The next one is Prevotella and Porphyromonas. So they are pigmented anaerobes. Porphyromonas melangenica, it is black or brown color colonies due to hemine derivative, and colonies and even dressings from wounds infected with this bacillus gives a characteristic red fluorescence when exposed to UV light. Now gram-positive anaerobic rods. So one is Bifidobacterium. So they are pleomorphic bacilli showing true and false branching. Then next one is Lactobacillus. So they are also called as Doderlin's bacilli. So they are normal flora in mouth, intestine and adult vagina. Then next one is Mobiluncus. It is motile curved gram-variable bacilli. There are some clues to anaerobic infection. So first is precipitating factors like diabetes, immunodeficiency state, then predisposing factors like ischemia, tumor or foreign body present in that infection, or foul odor of the specimen. Then they are typically polymicrobial. Then location of infection in proximity to mucosal surface. Then infection secondary to human or animal bite. Then gas is present in specimen and failure to respond to common antibiotics. Now we'll see lab diagnosis of non-sporing anaerobes. So specimen is pus, wound swab, aspirations under anaerobic precautions, sample collection done immediately in Robertson cooked meat media or thioglycolate broth to avoid exposure to oxygen. Then we will see macroscopic examination of the specimens caused by anaerobic infection. So they are foul odor, purulent appearance of fluid specimens, then presence of necrotic tissue and gas, sulfur granules present sometimes, then brick red fluorescence under UV light associated with pigmented Prevotella or Porphyromonas species. Then we will do gram staining, that is direct microscopic examination. So in that you can see pus cells and many microorganisms, that is polymicrobial. So you can do inoculation in blood agar plates, and that plates kept in anaerobic jar or gas bag system. We can do inoculation in Robertson cooked meat media. Identification of non-sporing anaerobes is done by biochemical reactions.
Hope you find this session useful for your understanding and for your exams. Thank you.